Snow day. Hope you guys are all nice and cozy with your socks on and no school. So we don't know if we're going to have school on Wednesday either, so we'll see. Fingers crossed, right? You want to have the midterm? Whoa, that was not supposed to happen. So I'm trying this new program. Oh no, I erased everything. I guess it erases that too. Anyway, I'm trying this new whiteboard program, so it's going to be a little weird. So let's get to it here. You can follow along with your midterm review sheet. I sent this to you guys. So really quick, the first unit that we had was basically how to run an experiment. A couple of things you have to know about experiment is the format for a hypothesis. Make sure that you know it's a statement, not a question. It doesn't have to be if and then. You might see something like increasing salt, increasing X will decrease blah Y. Okay, so that's just a prediction. You don't necessarily need a because, but you do have to make sure that it's in statement form, not a question. So anything you see that has a question mark is wrong. Data, anything that's measurable, and we, for the most part, will concentrate on quantitative data, which is numbers. Controlled experiment, 2 groups are needed. Control group, basically we need a control group so that we can actually compare our experimental results. So if we are measuring how plants grow with different solutions or different fertilizer, you'll take a normal plant, the control group, which is the one we use to compare. Okay, so that's called the control group. What else? Independent, dependent variable. So as temperature increases, evaporation, let's pretend, will increase. So that's basically the way it works is like this. If we write this sentence as temperature increases, evaporation will increase. These are the 2 variables. One is the independent and one is the dependent. The first one is always the independent variable. The second one is the dependent. And the independent, remember we said if you have a graph, it's the one that goes in the bottom. So the temperature is the independent variable and evaporation is the dependent variable. Now you have to understand lines like this. Oh my gosh, that's ugly. Sorry. You have to understand sort of relationship lines. So as temperature increases, evaporation will increase. And so that looks like that. So understand that. Oops. That's weird that it erases that. I don't like that. I don't like that. What else we have? We have presenting data. Okay, scatter plots. Basically, if you have, again, I'll use that temperature and we'll say, what could we use? We could use temperature and rate of molecule movement. Sure, why not? These are both numbers. So anytime you have numbers, oh my gosh, what was that? If you have numbers on either side, you're going to use a scatter plot. So that will look like this scatter plot with sort of like a trend line that goes through. So that's if there's numbers on either side. If you have categories, X, Y, Z, 3 different things, like we said, paper towels or brands or people, then we use a column graph or a bar graph like that. Okay. And then if you have time, that will be a line graph. So a time could be years or minutes, even though those are numbers, any kind of time could be, you could use a line. Okay. And then basically in the beginning of the year, we talked about what biology was all about and what makes things alive. So just remember the common things, like all living things are made up of cells. They adapt, they grow, develop and grow. They respond to stimuli. That means if you poke them, they'll probably respond somehow. And all of them contain genetic material from the smallest bacteria to us. Just to review what's alive real quick, because I don't have it in this review. Remember, we say that there's the 5 really kingdoms are bacteria, protists, fungi, plants, and animals. And what you need to kind of understand about each one is what kind of cells do they have. So bacteria have very simple cells. Those are called prokaryotic cells. And that means they have no nucleus. So if we were to draw a prokaryotic cell, it would just be like a bunch of DNA inside this little shell, okay? They're the only prokaryotic ones. Everyone here is eukaryotic. And that means these guys have cells that have a membrane enclosed nucleus like that, and they have a bunch of organelles that have like different little jobs. Fungi and plants have cell walls which surround which surround the cell. So if we put a brick like that around this guy, that would make it a plant cell or a fungi cell. And then we can sort of define how they eat or feed, or what kind of, so that's prokaryotic. This is eukaryotic. That's what the picture would look like. If you see anything with like a nucleus, then you know it's eukaryotic. And then we can say if they're autotrophic or heterotrophic. So don't worry so much about these top two, because they could be both autotrophic, heterotrophic, so that gets confusing. But fungi are heterotrophs. That means they have to eat. Plants are autotrophs. They use the sunlight for food. And animals are heterotrophs. Cool. So we got all that. So that's a nice little intro to everything that's alive and basically the types of cells they have, stuff like that. Remember, we also talked about specific animal groups, which that's porifera, cnidarian. There aren't too many questions on the midterm on that. I'll show you pictures of an animal and you kind of got to know what it is. Or I'll ask you who's related to the insect, and that's like another arthropod. So if I say insect, you say crustacean, something like that. Okay, so just review your animals. I won't review it in this little review tutorial. Okay, so that's Unit 1. Boom. Let's go to Unit 2. Unit 2 is your basic chemistry. So matter is basically anything that can take up space and has mass. What did I do here? I got to choose this. Anything that takes up space and has mass is matter. Okay. And matter can come in different forms. We said you can be an element. And if you're an element, you're on this periodic table down here. So you could be like oxygen, you could be hydrogen, etc. You're one of the elements. If you are a compound, you're two or more elements. So H2O is our famous compound. CO2, that's a compound. These guys are bonded together. These are covalent compounds because these electrons are sharing. And if you have like our famous salt, this is an ionic compound, which I'll get to in a second. So you have an element, you have a compound, and then you have a mixture. And so a mixture is basically like air. Because air has oxygen in it, has carbon dioxide, has water vapor, and so there's a bunch of stuff in air, okay? So that's considered a mixture. So that's basically matter. It's our chemistry basics. And remember, we talked about chem basics because we were leading up to how organic molecules bond. So we talked that there's elements, and we said that carbon was like our number one element in biology because it can share in 4 different directions, makes 4 covalent bonds, so you can have these like awesome chains. Let's see what else we have on our sheet here. Compounds, good, mixture, yeah, pizza, love it. Atoms, smallest unit of matter. Okay, so an atom, remember we said there's protons, which are positive, and neutrons, which have no charge. And we'll do 2 of each. Protons and neutrons are in the center, and then surrounding the nucleus are electrons, and electrons are negative like this. Okay, I shouldn't have drawn that other ring. There's nothing in that ring because we have 2 protons and 2 electrons to make this a neutral atom. Okay, if we had to know what element this is, we just look at the number of protons. There are 2, and that is here. That's the atomic number. This would be helium. Okay, so that's one thing you have to know. If you get a little periodic table, just know number. Oh no, how dare you! I don't know how to do that. Okay, I wonder if I can add another layer. I won't figure it out now. Okay, sorry, I get distracted. Protons are positive, neutrons are negative, electrons, atomic mass. the mass of a atom is totally based on, let me draw those two electrons again. Electrons are so tiny and insignificant, their mass, that they don't contribute to the overall mass of the atom. So when we're trying to figure out the atomic mass, we're basically just adding these protons and neutrons. So this guy kind of would have a mass of 4, okay? Now there's something called isotopes, and isotopes are having extra or less neutrons. And what that does, if you remember, is if I add another neutron here, that makes it heavier. So this guy now is 5. But the element doesn't change because it still has 2 protons, and 2 protons is always helium, okay? So isotopes, extra or less neutrons. Got that. Okay, ionic versus covalent. Just know covalent, I said, was sharing electrons. You don't have to know like any of this stuff, that we talked about, and the Lewis dot structures where we put dots on things. You don't need to know that for the midterm. You just have to know generally that covalent is sharing, and the reason that ions, this is an ion is any element that has a charge. Ions love each other because it's like a magnet. The opposite charge will hook up together, NaCl. This guy has no charge, no overall charge, because they cancel each other out. So salt has no charge. But as soon as you separate them, sodium has a charge and chlorine has a charge, and they're called ions. Now remember, the reason they have a charge is because they gain or lose electrons. So you have to understand kind of the stuff, the periodic table stuff. Like for example, here's chlorine, which I just used. That chlorine gained an extra electron because it wants to feel like a noble gas. These are the noble gases, and the noble gases have a filled outer shell. Okay, if you remember, the rule was 2 electrons like to be in the first ring. Here's the nucleus. 8 like to be in the second ring, and 8 like to be in the third ring. If for some weird reason, like chlorine, you have 2, and then you have 2, 8, and 7, that's chlorine, it wants that last little extra one to get 8. As soon as this little negative electron hooks up there, this guy gets a charge, okay? So that's how we get an ion. So just remember how to use your periodic table. Anyone who's in the column right before the noble gases is a negative. So fluorine is -1, Cl is -1. Anyone that's 2 away is going to be like 2 negative. Sulfur is 2 negative, you see? And so on and so forth. Nitrogen would be 3-, phosphorus would be 3-, okay? On this side, these guys become positive ions with these all being plus, so like K+, and these right here, these are the 2 famous ones, are like Mg2+. So that's how we remember our ions, gaining, losing electrons. Okay, that's it for that unit. Bam, the third one, organic molecules. Okay, so carbohydrates, remember, are starches and our sugars. Remember that if you see a hexagon, that's glucose most likely. It doesn't always have to be glucose. Like fructose is also a hexagon. But just know that this little shape is a simple sugar, a monosaccharide, okay? As soon as you put 2 of those hexagons together, and my art is fantastic, this is a disaccharide. And that's like this is table sugar. To save time, I will not try to spell. So this is disaccharide, which we have to digest, okay? And then polysaccharides. Polysaccharides are your long chains. So I had to draw a bunch of hexagons that are together. And our famous ones are starch and cellulose. Those are big, big, big polysaccharides, okay? So what else? So that's monosaccharide, disaccharide, polysaccharide. This one, remember, we have to break down because glucose likes to diffuse into our cells, and this polysaccharide is way too big. So we have to break all these bonds with digestion. What else? Let's see. Complex tube. Oh, I said that there. Great. Monosaccharides, disaccharides. I'm just reading your review sheet, even though I typed it. Oh, yeah. So one thing to understand is if you're doing sports, sugars are great for a burst of energy. So that's why in a marathon, you just take sugar packets or Gatorade. And then if you're the night before, you will eat starches like pastas and breads, which is stored fat. Okay. So that's your saccharides and your carbohydrates. Okay. Next is your lipids. Remember, lipids, we sort of have like, I'll draw the healthy one. This is the three tails. This is a glycerol head with the fatty acid tails, if you remember. And these like squiggly things, if you like look really close, it's just actually a bunch of carbons chained up like that. We just draw the diagram with these little squigglies. And what you need to remember is that if you have a fat molecule that has the carbon chains very straight, this guy is solid at room temperature because they're so tightly packed next to each other that they don't move. So you got your butter. And then this guy over here, they're so spread out, the molecules, that they glide past each other, and this guy's a liquid. So this one's liquid at room temperature, this one's a solid. The solid saturated, that's the one that's unhealthy, okay? Just remember, butter ain't good for you. Liquid, this one is the one that's unsaturated, and this one's the healthy one. And remember, the other dumb way we remember that is if the molecule's bent, you're like totally an active person. You're going up hills. And then if the molecule's straight, you love flat, unhealthy, non-exercise roads. So healthy, unhealthy, unsaturated liquid, and olive oil is sort of our famous example. Olive oil, nuts have this kind of fat, yada, yada. What else? Unsaturated polypeptides. Okay, off to proteins. And then what I'll do is I'm going to actually stop the video after this one and then continue it so it's not too long. So polypeptides, these are your proteins, and they do a bunch of stuff. But just remember the following. Enzymes. Enzymes, remember, are little proteins that speed up reactions. Polypeptide is the name of a long chain of amino acids, like this. It's a polypeptide. And this polypeptide chain, which starts off as a long train, remember, starts to wrap it into an awesome shape like a ribbon. And protein shape actually determines its function. So that's why it's important that they keep a perfect shape. If you heat them up, remember, they get denatured and messed up. And so they will cease to function. So polypeptide is what this long chain is called. These are called peptide bonds. What else? Enzymes are proteins. Antibodies, transport. You don't need to know this. Transport, cell membrane, enzymes. The word catalyst, you don't need to know. Proteins are made in the ribosomes. Okay, so remember our transcription translation that in a ribosome, you basically are using tRNA. I'll get to that later, but you use tRNA, remember, to build up the polypeptide. So this is all happening, remember, in a ribosome, okay? And last but not least, nucleic acids. Nucleic acids are just DNA and RNA, and I'll quickly talk about the two. One difference, DNA, RNA. DNA is double-stranded, and RNA is single-stranded. DNA has ATCG as the nitrogenous bases, but RNA has AUCG. They both have a sugar, and we draw that. Remember this? Oops, I'll draw the little flag there, okay? A little flag and a little circle. That's horrible drawing, I'm so sorry, gosh. It's like usually drawn as a little Lego piece here, okay? That's totally worse. This is the sugar, the phosphate, and the nitrogenous base. And what this is one little piece of DNA. So if you have the whole ribbon of DNA like this little brick guy is just like one, boop, boop, boop. It's just a small little piece in there that you can totally zoom in and look at this molecule. So it's got a sugar, a phosphate, a nitrogenous base. And this sugar is called deoxyribose. And RNA sugar is ribose. Okay, so that's the difference between them. Remember, DNA is the code, the genetic code, which is your gene. Okay, it codes for a specific gene. And RNA is the one that actually makes, helps make the protein with its tRNA and mRNA carrying the signal, which we'll get to more in detail when we go over genetics in our next video. Okay, I'll stop there because it's like 20 minutes long and I'm babbly. All right, love you. Stay warm.